All right. So I want to look at some more things that we can see from this mountaintop where we are. Now that we have learned about the basics of what's now called molecular biology, DNA, RNA, proteins, transcription, and translation. I've mentioned several times the fact that we can sequence DNA. We can read what the order of nucleotides is in any given piece of DNA, whether it's virus DNA, like the DNA of the coronavirus that's been sequenced and enables us to look for mutations, or the DNA of anything else. If anybody is sufficiently curious, you don't really have to do this, but there is an international database maintained in the US by the National Center for Biotechnology Information, which is part of the National Institutes of Health. It's called GenBank, and it has a ridiculously large number of DNA sequences that have been isolated from, I don't know, hundreds of thousands of different species of animals, plants, fungi, protists, bacteria, archaea, and viruses. And you might well be wondering how we're able to do that, how we can read any DNA when it's far too small to actually see. You know, I've talked a lot about how the order of bases on a DNA strand is critical because that carries information, but how do we know what the information is? To find out, we have to look at the other great scientist who got his PhD from the University of California at Berkeley. You already know me, actually quite a lot more people than just two have, but this guy in particular is biochemist and surfer dude, Dr. Kary Mullis. Back in the 80s, he was surfing quite a lot. And in his time not spent surfing and not taking LSD, he was working for a biotech corporation in the San Francisco area called Cetus Corporation. I'm not sure if they're still a going concern or if they got bought and merged and bought and merged. And in 1985, after taking a rather large dose of LSD, he got the idea for a technique by which he could take any piece of DNA and make millions of exact copies of it. I said way back when that science that science may seem uncreative to people who've never done it, but in fact, coming up with a really good hypothesis or a really good test of a hypothesis is a profoundly creative act. I know I said that earlier, and a good hypothesis can be, as rare and as beautiful as a good baseline. And just as some musicians have occasionally used chemical methods to come up with really good baselines or songs or things like that, there are some scientists, and he's not the only one, that have used recreational chemicals to come up with hypotheses. I'm not recommending it, but people have done it. And to be fair, Mullis was working as part of a team. If you're working for a biotech company, you don't work alone. The stereotype of the lonely researcher up late at night in his lab working on some earth-shattering discovery that only he can understand is not the way that most scientists work. He was working as part of a team, and there were other people that had come up with similar ideas, but Mullis was the prime mover in making the idea work, in pushing it through to a place where it was practical. And he ended up sharing the Nobel Prize in Chemistry in 1993. Incidentally, he went on to write this autobiography, Dancing Naked in the Minefield. He is a very quirky guy. hang on, it's telling me my connection is unstable. Where was I? We had a little instability there, but Mullis won the Nobel Prize. He's a very quirky person with some rather strange ideas. Certainly not a conventional scientist at all, but he certainly did make one really big score. And what Mullis and company came up with was the polymerase chain reaction, often abbreviated as PCR. You'll see this referenced, especially if you watch TV shows with a lot of forensics, you know, NCIS or CSI or something like that. But what exactly is PCR? Okay, you might remember way back when I mentioned something called extremophiles, and those are archaea or bacteria that live in environments that are too extreme for most other forms of life. Environments that are too hot or too cold or too acid or too alkaline, et cetera, et cetera. Now, I mentioned that there were plenty of archaeans that were extremophiles, although certainly not all, but there are some true bacteria that are also extremophiles, and this is one of them. This is a long rod-shaped bacterium, aka a bacillus, and it happens to be called Thermus aquaticus. And, all right, just got distracted because I have to grab a cat and keep her from tap dancing on my keys. Remember that the final is going to be cumulative, and a number of details that I mentioned back in the second part or even the very beginning of this course are going to dovetail with things that I'm bringing up now. There was a reason for me to bring up a lot of these. So make sure you go back and, you know, if you want a good grade on the final, now would be a really good time to start studying the older material with an eye towards how it fits into the new stuff that we're covering. You know, knowledge is connected. You know, all knowledge is ultimately one. We can't really split it up into little blocks, although we kind of have to pretend to in order to teach a course. Anyhow, so this is the extremophile bacterium, Thermus aquaticus, and like all organisms, it produces enzymes that replicate DNA called DNA polymerase. Now, all critters make DNA polymerase, but here's the thing. At very high temperatures, proteins, if you remember, may change their shape and lose their ability to function. They lose the possibility of doing whatever it is they do. And I've mentioned this before, we call it denaturing. So if you heat egg white, that's very rich in proteins, and the proteins go from being liquid and gooey and translucent to solid and opaque white. And you've seen this every time you have cooked an egg or seen one cooked. That's denaturing. The reason I bring that up is that most organisms can't survive at high temperatures because that high temperature will denature their proteins. If you jumped into water at about 56 Celsius, which is what, like 150 Fahrenheit or something like that, that's hot enough to start giving you severe burns. And that's hot enough to kill you. And part of the reason why extreme heat will kill you is that if it keeps up, your proteins start denaturing and lose the ability to do what they're supposed to do. DNA polymerase from Thermus aquaticus, however, is adapted to tolerate conditions like that. DNA polymerase from Thermus aquaticus is able to work under high temperatures, the temperatures where Thermus aquaticus lives. So PCR works like this. Basically, you heat DNA, and if you take a piece of DNA and you heat it, the hydrogen bonds that hold the two strands together with each other break, and the DNA unzips. Now, this happens every time you duplicate DNA in your own cells. And in that case, you have enzymes that do it, but DNA will also spontaneously unzip when you heat it. So you heat DNA, it unzips, and then you add the DNA polymerase and some nucleotides. And at that high temperature, the DNA polymerase will build new strands. Then you cool your DNA so that the strands come together and you have more double helix shaped molecules. And then you heat it again. It unzips, new strands get built by the DNA polymerase. Then you cool it and you heat it again, and you keep cycling it through. You heat it, you cool it, you heat it, you cool it, and you repeat this until you've got millions of copies of whatever DNA you started with. There's a little more to it than that, but that's the basic idea. By the way, this psychedelic thing over on the left-hand side, that's a computer model of Thermus aquaticus DNA polymerase. You can see it's got lots of alpha helices in it. You can make a neat little modification of this procedure, and I don't really have time to get into what it is. It's fairly simple, but that makes it possible to directly sequence DNA. You can take a single piece of DNA, copy it millions of times, add a little modification.
It wasn't working. I couldn't figure out why.
Mute Levi,
I just
please.
had to call you on my personal phone for some reason.
Levi, could you
Anyway.
mute? Okay, great. Okay, you can take any given piece of DNA, copy it millions of times.
Okay, that's why. In the email, it was 903.
Mute, Levi.
Yes. That's okay. I saved your number, so.
Mute. I can't really do the, I can't mute him myself when I'm in this full screen mode.
I have built the queries just by like.
All right, one
sections
moment.
of Arkansas, so yes, but nothing relevant to the school district.
Okay, hang on. I'm trying to find him.
SW4.
All right, there's that. Okay, so with this modification, you can copy a piece of DNA, do gel electrophoresis of fragments of DNA, each fragment of which ends with a different nucleotide, and read the order of nucleotides much the way that you'd read notes in a musical score. So you read this, in this case, you would read it from right to left, sorry, left to right, and each one of these little black bars marks the position of a nucleotide. So this would be G, G, C, T, T, C, G, A, G, 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 A, C, T, A, C, A, G, 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 and so on. In fact, if you wanted to, you could assign a pitch to each one of these nucleotides and you could sing it. You know, you could sing like, you know, G, G, C, T, T, C, G, A, G, 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 A, C, T, A, C, A, G, 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 A, G, C, T. Okay, it's kind of weird because the names of the notes are not the same as the names of the nucleotides, but there have been musicians that have experimented with turning DNA sequences into melodies. You can do things like that if you want to sing somebody's DNA. That's for the music majors here. And that's what makes it possible to identify DNA samples from individual people, even if all you've got is the tiniest little amount of DNA. In principle, if everything goes well, which it doesn't always do, you could take a single hair follicle or smear of blood from somebody, maybe a crime scene or some other sort of evidence, and duplicate that piece of DNA millions of times over. There's a little added refinement to make sure you're duplicating the right part of DNA that I'm not going to have time to get into. And then you could, if you needed to, sequence that DNA and read it. And since everybody's DNA is just a little bit different, you can look for differences that enable you to pick out one individual out of many possible ones. And the techniques that are used are called DNA fingerprinting. There's more than one way to do this, and Abby knows them all. We're not going to have time to discuss all of them here, but there's various ways this can be done. And the most common method takes advantage of the fact that everybody has what are called short tandem repeat sections of their chromosomes, or STRs. Not all of your DNA consists of genes, of parts that actively code for proteins. Some of your DNA consists of what we call promoter sequences that are involved in controlling when genes get transcribed and translated. They're involved in regulating what we call gene expression. There are some that have other functions. There's some I don't think we're entirely sure about. And there's some DNA that does nothing. And in STRs, the nucleotides don't normally make up a gene, although there are some cases where they might be part of a gene. But the nucleotides just repeat a short sequence over and over. So you might have a part of your DNA just goes CATG, 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 and so on. So you might repeat anywhere from about 3 to 6 nucleotides over and over and over. And these are very variable parts of your DNA because your STRs vary in length a great deal among people. So at one portion of my DNA, I might have CATG repeated 7 times, and you might have it 13 times, or 5 times, or 9 times, or whatever the case might be. And normally this doesn't have any effect on you. There are some genes where having too many STRs can disrupt their function. If memory serves, Huntington's disease is one of them. But for the ones that we're going to talk about here, these don't do anything. It doesn't make any clinical difference that I might have 9 of these repeats and you might have 12. It's just a difference. But we can isolate the parts of DNA that contain these short tandem repeats, take a sample, copy the DNA using PCR, and then use gel electrophoresis, which you've already done in lab, and we use that to measure the length of DNA fragments, right? Short pieces of DNA travel faster through the gel than large, so you can sort out pieces of DNA by size by doing gel electrophoresis. That's what you should have all done. And we can compare the STR lengths in an unknown sample with the STR lengths in a known sample. Okay, so there's gel electrophoresis again. In case anybody didn't get to do it last week, you put samples of DNA fragments into these little wells in a gel, which is a slab of what feels like kind of like Jell-O, made of a carbohydrate called agarose. And you then apply an electrical current to the gel. And since DNA has a slight electrical charge, DNA is attracted to one end of the gel by the electric field that you put it in. And small fragments of DNA pass through the gel more quickly. Large fragments of DNA pass through the gel more slowly. And the upshot is that you sort DNA fragments by size. And what you get is these bands, each one of which corresponds to a bunch of DNA fragments of a particular size that are moving at a particular speed through that gel. Okay, that should be very familiar to everybody by now. For any single STR, 5 to 20% of the human population will have the same length. Like at one part of my, in one particular part of my DNA, I might have 9 repeats of that CATG. And, you know, for all I know, Kim Jong-un might have 9 repeats of that same STR, and it doesn't mean anything. You know, it's, you know, that's purely chance. However, there's thousands of STRs in human DNA, not all of them equally useful. But if we were to compare the size of 6 or 8 or more STRs, it starts getting much less likely that 2 different people will have exactly the same lengths of all of them. The FBI's combined DNA index system, when you get your DNA taken as part of a crime investigation, the data gets stored in this giant database called CODIS. And originally it stored data on the length of 13 STRs. More recently, they've started storing data on 7 more. And the odds of 2 people who are not related to each other having 13 STRs match up perfectly is 1 in 1 quintillion. 1 out of 1 followed by 15 zeros. Which is far bigger than the population of Earth. That's far bigger than the total number of people that have ever lived. So if somebody isolates DNA from a crime scene and they calculate the size of 13 STRs, and they find that my DNA matches all 13 STRs, then the odds that it wasn't me are very, very low. I mean, it might still be possible, but it's extremely unlikely that that DNA belongs to anybody else. And this happens to be a map of the 13 STRs that are used by CODIS. As I mentioned, there's over 10,000 STRs. Most of them are not especially useful for various reasons. These are the 13. There's one located, each one of these bar diagrams right here is a map of a chromosome. So there's a site on chromosome 2 that just happens to be called TPOX, and that's one of these STRs. You know, I always thought Tupac was the guy that had the famous beef with Biggie Smalls, but I guess not. There is a location on chromosome 3 that happens to be called D3S1358. That's another STR. That's another one of these places where people have different numbers of repeats of some short DNA motif. And there's one on chromosome 4 that's called FGA. And there's one on chromosome 5 that's called D5S818. And another one that's called CSF1PO. That's C3PO's uncle, and so on. And all that these codes represent are they're just codes for locations in human DNA that have these repeats. And these are the 13 that are routinely measured and stored and used for identification. And this is a part of what an individual's DNA fingerprint looks like. So right here, this says D19S433. That's just the name of one of those tandem repeats, and it's on chromosome 19. So this guy has 2 copies of chromosome 19, one, of course, from mom and one from dad. And one of his copies has a certain STR that's repeated 13 times. And the other chromosome has the same STR, and it happens to be repeated 14 times. And then here, VWA is another one of these STRs. It happens to be on chromosome 12. And this guy's got 2 copies of chromosome 12, one from mom and one from dad. And one of his copies of chromosome 12 has this STR that's got 14 repeats. And his other chromosome 12, the same place, happens to have 17 repeats. And then over here, this is TPOX. That's yet another STR on chromosome 2. And in this case, both of his chromosomes 2 have 11 repeats of the TPOX repeat, and so on. That's what an individual's DNA fingerprint looks like. Now, we've talked about forensics. When this technology became available, there were some pretty serious worries about privacy issues. There certainly are ways that DNA evidence can be misused. The idea being that we all shed our DNA into the environment all the time. There's DNA in the follicles of every hair that falls off your head and goes down the shower drain. There's DNA in the flakes of skin that we're all shedding. There's DNA in every drop of saliva, that you might leave on a glass that you've drunk from. And DNA analysis, PCR, is so sensitive that even the tiniest trace of DNA can be picked up and sequenced. So there was a lot of worry that basically, since we're all unavoidably giving away pieces of our DNA all the time, that the government was going to, sequence all of this and, be able to track our movements and things like that. There certainly are ways that that could happen, that evidence can be misused. And like everything else, you have to weigh individual privacy against societal need, which is a much bigger conversation than I'm prepared to have. But one thing I'll point out to the good is that DNA evidence has also been used to overturn wrongful convictions. As of, I don't have the numbers for most of 2021, but as of April of last year, 375 prisoners had been exonerated by DNA testing. They'd gone to prison for a crime. Law enforcement had finally, usually after lawyers ragging on them to do it, had finally tested physical evidence from the crime, maybe blood or semen or saliva or something like that. And it had been found that, the blood or semen or saliva or something else found at the crime scene did not come from the person who'd been locked up in prison. 21 of the prisoners that have been exonerated were on death row. These gentlemen here in the middle, Levon Brooks and Kennedy Brewer, had been in prison for years for rapes and murders of children, but freed in 2008 when DNA testing showed that they could not have been the people that committed the crimes. It is unjust that they were locked up for this long. And in some cases, they were locked up on the basis of shoddy or dodgy police work. But even if we can't undo that, at least it's been possible to give freedom to people who've been unjustly convicted. So DNA technology, like everything, is a potential double-edged sword. There are ways it could be misused, but a positive way that I'd kind of rather think about is using it to, using it to increase justice and get people out of prison when they've been unjustly imprisoned. A lot of this, by the way, has been done through a nonprofit called the Innocence Project, on whose website you can read more details. That's actually the end of the slides. I don't really have anything more for you today, so I'm going to go ahead and stop right there, and I will now stop the recording.